This will be recorded and it will be shared publicly. So mind yourself. Okay, I'll take a few minutes here to introduce Anna, and then I will pass the baton over to Anna to give her talk. So first off, Anna Phan is one of the newest members of the Department of Biological Sciences. She arrived earlier this year. I believe, Anna, and you correct me if I'm wrong, your hometown is Toronto.
Correct, yes.
Yeah, okay, right, so another Torontonian. Anna did her first degree, her honors degree at the University of Waterloo, where she did actually her honors project in the lab of some character named Brian Dixon, who's an old friend of mine, it would turn out. Brian was the senior graduate student when I was a junior graduate student at Dalhousie, and Brian and I spent a lot of time together. We played ball hockey together on the weekends. We drank beer together on the weekends, and he showed me how to clone and a whole bunch of other stuff. So shout out to Brian Dixon, who's one of my absolute favorite people. So it's interesting how small the world is.
Yeah.
And you worked on, I guess you did some sequencing, some phylogenetics of char MHC. So it's some fish immunogenetics. And after that, Anna went to the University of Guelph, where she started working in neuroscience, and she did an MSc and her PhD at Guelph. Following her time at Guelph, she became a postdoc in neuroscience again at the Scripps Research Institute in Jupiter, Florida. Was there for some time until earlier this year, when around the time that COVID hit, she decided to pack up and move to Canada, move to Alberta, and I guess that was a bit of an ordeal, right? Getting across the United States and across the border, right at the peak of the outbreak. And when was it? Was it in April when you arrived?
Yeah, just at the beginning of April.
Just at the beginning of April. So, like, yeah, it was a pretty crazy time, and we're delighted that you were able to arrive here safe and sound, and spend your 2 weeks in isolation in a hotel on the south side of Edmonton, I think, before you could actually, come to work.
Yeah.
And you still can't really come to work.
Yes.
But welcome to Edmonton. Welcome to the University of Alberta. Okay, with that, I'm gonna pass over to Anna, and Anna will share her screen, and away we go with your presentation. And I don't have your flyer up in front of me, so I'm gonna have to let you read your own title.
Sure.
Sorry for that.
No problem.
Okay, away we go.
Okay. Thanks, Dave. Let's share my screen. Okay. Okay, thanks everybody for joining me today. I gotta just keep an eye on the time so I don't go for too long. So the title of my talk today is Memory Suppressor Genes Reveal Constraints on Synaptic Vesicles. And I also realized earlier today that this is the first talk that I'm giving as an assistant professor. So yeah, that was fun. And I guess the other interesting tidbit about starting a position in the middle of a pandemic is that I have yet to meet a lot of people in the department. So I hope that one day I will get to meet other people that work in this building. And so, yeah, I'm looking forward to hearing the other speakers in this chair's lecture series over the year. But I'll get started on mine. So I'm a neuroscientist, and my primary interest is the neurobiology of learning and memory. My primary interest is in the neurobiology of learning and memory. And really, learning and memory is, at its core, the ability of the nervous system to record new information, to store that information for a period of time, and then retrieve that information. So, arguably, a large part of the neurobiology of learning and memory actually involves studying the genetics of learning and memory. Because through studying the genes that are involved in promoting learning and memory processes, we're also able to learn about what cellular mechanisms are necessary for these processes, etc. And probably it comes as no surprise that the topic of learning and memory is a fairly inherently interesting topic for many researchers in the field of neuroscience. And over the last probably 60 to 70 years, there has been quite an intense amount of research into identifying the genes that are necessary for supporting normal learning and memory. And this work has identified hundreds, it's more likely that there are thousands of genes that have been identified that are necessary for normal learning and memory. And this may sound like a lot, but I think it also speaks to exactly how complicated this process is. Because the genes that are involved in this are involved both in the establishment of proper neural circuitry as well as actively supporting learning and memory cellular processes. What you may not realize is that in addition to all of these genes that support memory processes, there's also a flip side of the coin. And that is that there are genes that exist to limit learning and memory processes in organisms. And these are referred to as memory suppressor genes in the field. In comparison to the genes that support memory, very little is known about the identity and the mechanisms of these memory suppressor genes. And in general, there have been about a few dozen genes that have been identified in this category. And so the focus of my talk today really starts with the identification of a lot of novel memory suppressor genes in Drosophila. And when studying, a phenomenon that's, I guess, still in its infancy in terms of its research, Drosophila, I would argue, is a really great model organism to use in this respect. One of the major advantages of this organism is the ability to perform genetic and RNA-sequencing screens in this using this model organism. And to this, there have been actually multiple RNA-sequencing libraries that have been developed in different stock centers across the world, which means that these RNA-sequencing lines are available to researchers. Just basically, you just have to put it in order and they'll send them to you. But there are RNA-sequencing lines available for over 90% of the fly genome. And this really speeds up the ability to do research, when you're working with genes, especially of genes of unknown function. And this is probably included in the fact that Drosophila has a very expansive genetic toolbox. But this is also one of the major advantages of Drosophila for neuroscience research. And one of the tools that I just want to mention very briefly is the GAL4 UAS binary expression system, which is really a workhorse in Drosophila research. So essentially, what this system allows us to do is to express genes of interest, for example, or RNAs in a very tissue-specific manner. And so as an example of this, this particular this particular GAL4 line that's named R89E11, that's not important, is expressing a green fluorescent protein in a large, expansive region of the Drosophila brain. Whereas this particular GAL4 driver expresses only in a specific subset of neurons that make up this donut-shaped structure in the fly brain known as the ellipsoid body. And this is really just to illustrate that we use this GAL4 UAS system all the time to manipulate, to express genes of interest or knock down genes using RNAi methodologies in very specific neuronal subsets. And this is very important to do when you're studying neurobiology, because depending on the type of neurons that you are manipulating, you can have very different outcomes, behavioral outcomes, neural activity outcomes, etc. And so just to illustrate this with a very simplified example, using excitatory neurons versus inhibitory neurons. So the same manipulation, if you increase the activity of excitatory neurons, leads to increased activity of downstream neurons in the circuit. Whereas if you increase the activity of inhibitory neurons, you actually have the opposite effect in that you silence or reduce the activity of downstream neurons. So I hope that through this example, you understand how important it is that we are manipulating our genes of interest and the activity of neurons in very specific neuronal populations. And this is really key to figure out how it is that specific genes are affecting learning and memory. Okay, and so this story really starts with this paper here that was published in 2015. And this came out of Ron Davis's lab at the Scripps Research Institute in Florida, where I did my postdoctoral studies. And this was a project which took advantage of all of those genetic tools that were available in Drosophila, and they performed an RNAi screen for learning and memory to identify new genes that were involved in the learning and memory processes. So from this screen, they looked at 3,200 RNAi lines, which is only a fraction of the fly genome. It's not even close to covering all the genes that are there. But from this screen, they found about more than 500 genes that resulted, when you knock down their function or when you reduce their function, resulted in impairments in learning and memory. So these are the genes that support normal learning and memory processes. However, at the same time, they also found about 40 potentially novel memory suppressor genes, meaning that when you knock down their function, you actually had enhancements in memory performance in these flies. So this table is those 42 potentially novel memory suppressor genes that were identified from this screen. And highlighted in yellow are the genes that my colleagues and friends either have worked with and published, or the work is still in progress. But I hope, so when we consider the genes for which the data has already been published, we see that their mechanisms of action are really quite varied. So for example, scribbled up here, it was shown to be important as a signaling as part of the signaling cascade for active forgetting, whereas CG7442 was shown to alter cholinergic neurotransmission, whereas RAS85D was shown to be a molecular switch between different forms of memory in Drosophila. And so I think this argues for, this is evidence for the fact that really memory, we should be studying and identifying novel memory suppressor genes in order to be able to understand more of the fundamental processes of how learning and memory works in general. And so the particular gene that I ended up working with was Stromalin, and so this is this will be the focus of my talk today. And so Stromalin, as it turns out, is a member of the Cohesin complex, which is a highly conserved set of proteins, but they're most famous for their role in maintaining proper chromosome segregation during cell division. And they're also known for regulating gene expression postmitotically. However, the role of Stromalin and cohesin complex or other cohesin complex proteins in learning and memory was unknown at the time I started this project. Okay, so if you are wondering exactly how it is that we study memory in Drosophila, most of the time we use what's known as the aversive olfactory conditioning paradigm. So conditioning is one type of associative memory, and it's probably for the public, it's probably best known through Pavlov's dogs, in which he trained dogs to associate the ringing of a bell with the presentation of a food stimulus. And so the presenting dog with food would cause the dog to start salivating, but with enough pairings of the bell and the food, the dog would learn to associate the ringing of the bell with the food stimulus and would start salivating upon ringing of the bell only without presentation of the food stimulus, meaning that they learned to associate the sound of the bell with the presentation of the food. And so in flies, we use the same type of learning paradigm, only we are teaching them the association of a specific odor with an electric shock stimulus. So during the training phase, we present the flies with one odor, in this case, odor A, in the presence of an electric foot shock stimulus. After which they're presented with a second odor, odor B, with no electric shock. These flies are then given a choice test in a T-maze between the odor A arm and the odor B arm. If they remember this association during training, they will avoid the odor A arm in favor of the odor B arm. And essentially what we do is we count the numbers of flies in each of these arms to obtain memory score. And higher values of the memory score indicate better performance on this task and hence better memory for this association. And we know, decades of work in this field of research have revealed some of the basics of the aversive olfactory memory circuit in the fly. And so this is just to orient you in the fly head, in the fly brain, there are these structures that we know as the mushroom body lobes that are illustrated in yellow over here. So the flies sense odors through their antennae here, and that odor information is carried via a couple of neurons to the mushroom body neurons in yellow here. We also know that the shock or the aversive information is conveyed through a particular set of dopaminergic neurons, shown in orange here, that connect to the mushroom body neurons along this vertical stalk of the mushroom body lobes. And therefore, this vertical stalk becomes the site of convergence of both the olfactory information as well as the aversive information, and therefore is an important site for aversive olfactory memory formation and retention. Output from these regions then are known to influence avoidance behavior in the flies. And because most people know dopamine as kind of like being more associated with rewarding signals, I just wanted to mention that this is also true in Drosophila. There is a separate set of dopaminergic neurons that synapse along areas of this horizontal lobe here, and that are necessary for forming appetitive memories, and output from these regions is known to influence or to modulate approach behavior in the flies. The paradigm that I'm going to be focusing on, and the neural circuitry that I'm going to be focusing on, is this aversive circuitry here. And so, like I said, from that RNA-sequencing screen that was published in 2015, Stromalin was one of the novel memory suppressor genes to come out of there. And so this is basically the starting point of this story. You can see that when we express stromalin RNAi to reduce expression of stromalin in all neurons, that these flies then exhibited higher memory scores compared to controls. And so my project and my job really was to, one, characterize these effects, and two, to figure out exactly how the cellular mechanisms behind how it is that stromalin was enhancing memory. Okay, and so the first step that we undertook was to identify in which neurons stromalin was having its effects. And we did this using the GAL4 UAS system to express RNAi in various subsets of neurons that were known to be involved in the aversive olfactory memory circuit. In doing so, we find that, when we knock down stromalin in all neurons, or in mushroom body neurons, or in dopamine neurons, we can get this enhanced memory effect in the flies. We chose to focus on stromalin's effects in the dopaminergic neurons here. So for the rest of my talk, I'm focusing on stromalin's effects specifically within the dopaminergic neurons here. Okay. The next big question that we needed to answer was whether this was a developmental or an adult effect of stromalin knockdown. And this is important to distinguish because conceptually, these are two very different mechanisms through which memory suppressor genes may work. So if a gene works through altering neural development, you can imagine that they might have effects on proper neural circuit establishment, versus if they had, if they affected memory during adulthood, then this would indicate that they played an active role, some active role in the learning or memory processes. So to distinguish between these two modes of action, we use another really nice genetic tool that's available for Drosophila, and that is, it's called Target, But essentially, it allows us to control transgene expression through altering the incubation temperature of the flies. So at low temperatures, for example, at 18 degrees Celsius here, if we raise the flies at 18 degrees, there is no transgene expression, so there's no knockdown, and we don't see the enhanced memory effect here in these adult flies. Oops. Sorry. Whereas if we raise the flies at higher temperatures, so at 28 degrees, we do have RNAi expression, and we do replicate the effects that we saw previously in that Stromalin knockdown flies display higher memory scores than controls. So these two groups are actually the control groups for this experiment, and these are the experimental groups. What we found was, very interestingly, is that Stromalin knockdown during development was necessary for this effect, and not during adulthood. This was interesting for a number of factors, mainly because this was the only known memory suppressor gene at the time to work during development to then enhance memory in adult flies. And it was also very difficult to imagine how it was that messing up some neurodevelopmental program could then result in flies that were better learners. So we kind of probed this, this result a little bit further, and we tried to pinpoint the exact developmental time point that was critical for Stromalin's effects on memory. And we did this by knocking down Stromalin in increasingly narrow developmental windows to try and figure out when that critical window was. And so through these experiments, we found that knocking down Stromalin during the third Instar larval stage was necessary and sufficient for the enhanced learning effects that we see in the adults. And in fact, you can knock down Stromalin throughout the life of the fly, but if you don't knock it down during this specific third Instar larval stage, you do not see the memory effect. What this told us is that Stromalin was somehow changing the function of the dopamine neurons during this developmental time window, and these changes then persist into the adult fly to then affect adult memory. So, so how exactly was Stromalin altering dopamine neuron function? As I said before, the dopamine neuron is responsible for conveying the shock or the aversive information in this particular learning paradigm. And we know that this, the dopamine neurons that innervate this heel region of the mushroom body played an important role for this, for the phenotypes that we were seeing. And we also know from prior literature that dopamine release onto these mushroom bodies or onto their downstream neuronal partner results in a cyclic AMP increase in the downstream neuron. And this cyclic AMP increase can actually be observed using an optical sensor called EPAC. So what we did was we used in vivo functional imaging to see how it was that Stromalin altered dopamine neuron function. We mounted the flies underneath the microscope, and then we delivered to them the electric shock stimulus that we would normally provide during the behavioral training session. And so these microscope images on the left-hand side just indicate where the heel region was located in this image. And EPAC is a FRET sensor, so we have two fluorescent proteins But I hope, but let's focus on the middle graph here. The red bars down here indicate when the electric foot shock was delivered to the fly. And the fly gets 12 90-volt electric foot shocks, which is the same stimulus they get during training for behavioral memory. I hope what's evident from this graph is that when you knock Stromalin down in the dopamine neurons, we actually see an increased cyclic AMP signal by its downstream partner. And this is increased compared to the cyclic AMP response in control flies in green down here. So this is consistent with the learning and memory effects that we see in these flies. And so the question then became like, why, like how it was that Stromalin was leading to this stronger sort of synaptic connection between itself and the mushroom body neurons. And so we tested several hypotheses that might explain this effect. So one, we tested whether the dopamine neurons themselves were more sensitive to the shock stimulus, and that was not the case. And two, because the cohesin complex is known to be involved in cell division, we looked to see if maybe we had more dopamine neurons. And that was resulting in more dopamine being released, and that's not the case. We also examined whether Stromalin knockdown resulted in increased branching of the dopamine neurons. So it might have like increased kind of fingers into the mushroom body lobes, and that might have been resulting in the increased cyclic AMP signal that we were seeing. But that also turned out not to be the case. Finally, we tried looking specifically at the dopaminergic synapses by using a presynaptic marker. So this was just a green fluorescent protein that was conjugated to a synaptic protein that localized to the synaptic terminals. And in doing so, what we found was the presynaptic marker levels were significantly higher in Stromalin knockdown neurons compared to control neurons. And so this was pretty exciting because this is consistent with the functional data that we had and the behavioral data that we were finding. And I finally had some morphological like correlate of these things, of the behavior that we were finding, and I was excited because this might also offer clues as to how, like, in terms of the cellular mechanisms of how this was occurring. And so we reasoned that if this increase in presynaptic marker levels was in fact important for the memory effects, that we should see these increases start to emerge during the third Instar developmental stage. And that's exactly what we see. So these increases in the presynaptic marker levels start during the middle of the third Instar larval stage, but prior to that, there's no differences. And so this gave us more confidence that these presynaptic marker increases were related to the behavioral phenotype that we were seeing. And so we couldn't stop there. Obviously, we had to go deeper. And so the question then became, well, what does it mean when we have increases in a presynaptic marker? We came up with 3 scenarios that we thought could reasonably lead to increases in presynaptic marker levels. And one was that it resulted in increased numbers of synapses. Two was that perhaps the size of the synapses were larger. And finally, because the particular marker that we were using localized to synaptic vesicles, that perhaps it increased the numbers of vesicles at the synaptic terminal. And synaptic vesicles, I guess you could think of them as information packages, essentially. And these information packets are what neurons use to communicate between each other. And so in order to kind of tease these scenarios apart, it wasn't an easy task, just because of technical limitations, really, for confocal microscopy. And so we had to turn to super resolution imaging, and in particular we used SIM super resolution, which is structured illumination microscopy. And essentially what it allows us to do is it allows us to get images that are with higher resolution than would normally allow for due to the diffraction limit of light. And so using this method, this is a new method, so it took a really long time to actually develop tissue processing protocols and tissue clearing protocols that we could use with this. But eventually we were able to get really nice images of the synaptic terminals in the whole fly brain. So this is again looking at the heel region, and these are the dopaminergic synaptic terminals here. And so after obtaining these images, we then used a image analysis software to reconstruct each of these particles in 3 dimensions in order to get measures of the volume of the particles as well as the numbers of synapses, dopaminergic synapses within this particular brain region. And after all of that, what we found was that Stromalin knockdown did not have any effect on the numbers of synapses, nor did it affect the volumes of dopaminergic synapses. Yeah, and so we could eliminate the first two hypotheses. And so we were left with, well, perhaps it might increase synaptic vesicle numbers. But in order to assay that, we needed to turn to electron microscopy. So we took serial sections and imaged those sections using the electron microscope, and we found the dopaminergic neuropil within the electron micrographs, and then proceeded to reconstruct in 3 dimensions the neural pill data as well as the location and numbers of all of the synaptic vesicles within the dopaminergic neurons. And hopefully what is clear from this 3D reconstructed image is that in the Stromalin knockdown neuron, we have a lot more synaptic vesicles than in the control neuron. So this is just that data quantified. We found that when we expressed Stromalin RNAi in the dopaminergic neurons, they had roughly twice the number of synaptic vesicles as well as dense core vesicles compared to control neurons. And using that 3-dimensional data, we also calculated the distance from each synaptic vesicle to its nearest active zone, which is the site of release of synaptic vesicles, to find that, in Stromalin knockdown neurons, we had about twice as many vesicles within 200 nanometers of an active zone compared to controls. So yeah. I always like this slide because this one slide really summarizes 4 or 5 years worth of work. And what we found was that Stromalin knockdown during a critical developmental window and the third Instar larval stage results in an increased number of synaptic vesicles and neurons. This then results in an increased synaptic connection with the dopamine neurons and its downstream partner, which underlies these flies' better learning abilities. Okay, and so that was the story that we published in 2019. And so there's still a lot of work that still needs to be done on this project. One of the things that I hadn't mentioned is that these findings with Stromalin are quite unique in that it appears that it's only synaptic vesicle numbers that are affected, but by all other measures that we have taken, and everything else about the neuron is normal. And so this really opens up the possibility of using this system to identify the genetic regulators of synaptic vesicle numbers, which is, I don't believe, a research area that anybody is pursuing at this moment. And our hypothesis, based on the data that we have, is that Stromalin, through its actions via the Cohesin complex in regulating gene transcription, is then what is regulating synaptic vesicle numbers. So for the last few years, I've been working to identify what the genetic regulators of the synaptic vesicle numbers are that are downstream of our Stromalin manipulation. To do so, I used RNA sequencing. And so what we did is we took the larvae of the flies from the mid-third instar larval stage, which is the first time point at which we start to see this effect. We get these larvae and then we dissect out their brains, and then we throw their brains in essentially a chemical cocktail that dissociates the cell bodies, and then we dump those in a petri dish. The dopamine neurons are labeled with a green fluorescent protein, and so we had to go in and manually pick the green fluorescent protein positive neurons, and we collected them, and we had about 25 neurons per sample, and we threw them into the RNA sequencing machine. In doing so, we found about 174, or exactly 174 genes that were significantly dysregulated with Stromalin knockdown compared to controls. And as you can see, some of those genes, some genes went up in expression and some genes went down in expression. Yeah. And so these 174 genes, of these 174 genes, 4 of these genes had no associated RNAi line available. And this is because these 4 were non-coding RNAs. And 5 out of these 174 genes, when I crossed these RNAi lines with the GAL4 driver, resulted in lethality. But the remaining 165 RNAi lines were then put through a primary screen in which we assessed the memory effects of knocking down these genes in the dopamine neurons. From this, we found about 53 RNAi lines that produced behavioral effects that were consistent with Stromalin's behavioral effects and the RNA sequencing effects. And so these 53 RNAi lines are now undergoing a secondary screen for synaptic vesicle effects. One of my 499 undergraduate research students, Chisholm, is helping me with this at the moment. And this is in progress, but we have already found 7 possible genetic regulators of synaptic vesicle numbers so far. And this is all very preliminary, so we need to go back and double check that the results that we got from the screen are accurate. And so we have a lot of work to do, but it seems quite promising thus far. Interestingly, as a result from this, we also found about 8 RNAi lines that actually look like they're potential novel memory suppressor genes. And the effect of these RNAi lines are such that they're opposite in the direction that we would expect if they were acting downstream of Stromalin. So this is also a really interesting finding, and 7 of these RNAi lines do not appear to act on synaptic vesicles, meaning that they might have other novel modes of action to limit learning and memory. So this is exciting, another aim that has kind of come out of this project. And so those are the first 2 aims for future directions I've already covered. But the last aim that I want to pursue is also to test if we can modulate synaptic vesicle numbers in dopamine neurons to rescue some of the Parkinson's disease-associated locomotor and memory impairments. So that's something that I'm just gearing up to get ready, or starting in the lab right now with a couple of other 499 students. Yeah. And so, yeah, this acknowledgement slide is getting kind of crowded. I keep updating it, but I'd like to thank, in particular, Molly from the Davis Lab, and Jacob Berry, who's also my husband and here now with me in the Department of Biological Sciences, for their help on this Stromalin story, as well as Naomi and Conan from the Max Planck Institute of Florida's electron microscopy core, who were instrumental in doing a lot of the EM, or getting a lot of the EM data. And also my former advisor, Ron Davis at the Scripps Research Institute in Florida. Okay. All right, questions?
Well, thank you, Anna. And I'd like to open the floor to questions. And this is probably a good time for folks who have questions to, well, you can use the chat or turn your camera on and wave your hand. There we go.
Oh, hi, Anna. Great talk. Thanks so much for that. I have a few questions, but I think I'll just stick to a couple.
Sure.
I'm wondering, so did you do anything to determine if there's actually an increase in the probability of release of transmitter? Because there are circumstances in which there are maybe no changes in any of the things that you might look at, but still there's an increase in the probability of release given the presynaptic stimulus. So that's the first part. And then the second part is, do you know if there's any change in the quantal content or in the actual vesicular content, vesicles, and the any type of sort of postsynaptic effect as well?
Mm-hmm. Yeah, they're all really great questions. So I guess the long story short is no, we don't know the answer to any of those questions. And so things like probability of release, as well as quantal content, we really need to we really need electrophysiological experiments to answer these questions, which we're not able to do in the particular neurons that we are looking at in the fly brain. We didn't have a rig in my postdoctoral lab. Although I've done electrophysiological recordings from mammalian neurons, but doing them from Drosophila neurons is even more challenging because of the size of the neurons, and it's just much more challenging. So we didn't actually have the ability to do that. Yeah, and so, those are interesting questions that remain to be answered. My guess, based on and this is just a guess, based on the cyclic AMP responses we see in the postsynaptic neuron, is likely not an effect of probability of release. So I don't know if I should go back to find the slide, but essentially what we see is that in the first few electric shock stimuli that we give, the first 3 or 4, there's very little difference between the control and the knockdown neurons responses. The differences between the 2 genotypes actually comes after repeated electrical stimulation. So I think that my guess is what that is indicating is that there is a larger pool of vesicles that are available there to keep up with sort of maintaining the same amount, maintaining dopamine release that is similar to the first initial response that we get. So it's just maintaining that it's able to keep up with that. So that's my guess. But yeah, really we need an electrophysiology rig to figure that out.
Okay, great, thank you.
I see Clayton Dixon's got his hand up. Clay?
Thanks, Anna. That's some really nice work. I really enjoyed your talk. I'd like to take a devil's advocate approach, perhaps as an electrophysiologist as well, and follow up on what Declan was asking. And I think I would probably go even further. So I would ask you whether or not the sort of manipulations that that you've done and shown here are actually producing activity-dependent differences in your knockdowns versus the non-knockdowns, which is then the result of the differences in terms of learning and memory that you report. And this is all my own bias, but
yeah. So what do you mean activity differences?
So what you've done is you've altered the neurophysiology of the system so that it promotes learning and memory, and it's a simple response, stimulus response function.
Yeah. So I guess one answer to that question is the fact that the dopamine neurons themselves are not more responsive to the electric shock stimulus. And so we measured that via GCAM. Again, because we don't have we didn't have the ability to do electrophysiological recordings. But in those experiments, it actually, if there was no statistical difference in the GCAM responses, in between the 2 genotypes of the dopamine neurons to shock. But if anything, it looked like the RNAi line had reduced calcium influx upon electrical stimulation. And so, yeah, so it's not that the dopamine neurons themselves were more sensitive to the shock stimuli. It appears to be really specific to the synaptic connection between the dopamine neurons and its postsynaptic partner. And then in terms of postsynaptic changes that are going on, I can't say for sure that it's possible that there are some postsynaptic changes that have gone on with our genetic manipulation.
Yeah, that's mainly what I was concerned with, not so much the dopaminergic neurons themselves and their responsiveness, but the postsynaptic neurons.
Yeah, but yeah, so, you know, I guess one point to consider is that we are only knocking down stromalin in those dopaminergic neurons. So we're not genetically manipulating the postsynaptic neuron. And that's not to say that you know, there still might be some postsynaptic effects, but at least our genetic manipulation is limited to the dopamine neurons. Mm-hmm.
Great, thanks.
Thanks.
Thank you, Clayton. Anna, had you met Clayton before?
No, I don't believe so.
Clayton's in the Department of Psychology, just to give you guys an introduction. So, you know, welcome, Clayton. I'm delighted to see you here, and thank you for participating.
Thanks for that. Well, I've met Jacob, but sorry, and I haven't met you yet in person.
Does anybody else have any questions for Anna? I see Ted's hand. Ted, the floor is yours.
Hey, Anna, that was a great talk. I enjoyed it. I just had a curiosity. I think you've talked about memory, you know, learning suppressing genes. I guess I'm wondering about learning suppressing genes versus memory suppressing genes. I know that some folks make an important distinction between the two, and you use your words carefully. So I was curious about why that was, and if there is such a thing as both.
Yeah, sorry. So I didn't go into this in detail just for time restrictions, just because of time restrictions. But we are using the aversive olfactory conditioning paradigm. We are able to test for, you know, learning suppression effects versus memory suppression effects. And so what we found is that this effect of stromalin is actually on learning, on memory acquisition, and not on memory retention. So that's not to say, like, so maybe I should describe the experiment. So when we assess their memory immediately after training, we vary the training protocol. We vary the number of shock and odor associations that are presented to the fly. So under normal circumstances, the 12 shock protocol, it produces ceiling levels of performance in the flies. But we also reduce that training protocol to see kind of their acquisition curve. And Stromalin knockdown flies are better learners. And when we train the flies in the normal, like 12 shock training protocol, and then check their memory retention, so i.e. just how well they retain that information over time after their training protocol, we see that they also have enhanced memory scores. But you can, but those enhanced memory scores can be secondary to the learning enhancements that we have. So we can actually tease those two apart by essentially correcting for the differences in acquisition between the two groups. So I trained the Stromalin knockdown flies with half the number of shocks that were given to controls. And when you do that, their memory retention scores afterwards are identical. So suggesting that, you know, the memory retention increases that we see are secondary to the learning enhancement effects that we have. So Stromalin actually is a learning enhancer, not a memory enhancer. But I know I mixed those two up because it's largely because I didn't go into it in detail during the talk. So
if you don't mind a quick follow-up. So it's amazing that you can tease those two apart using your system. I'm having trouble thinking through off the top of my head this sort of question about adaptive value. So adaptive value of suppressing learning versus adaptive value of suppressing memory. Is that something you guys are sort of have thought through?
No. So yeah, so in terms of the differences between the two, no, we haven't really thought of that. And in fact,
cool.
we don't even really know what the adaptive value of having memory suppressors at all is at the moment. And so that's sort of one of the questions I think that we should explore. And so some of the ideas that are floating around, and there's some evidence for these, is that if you have organisms that are like, that are too good at learning or have too good of a memory, that they're more likely to like kind of associate things that are not related to each other. And then there's also other ideas that it prevents generalization of learning and memory. Also, it might lead to other impairments and other types of behavior. But so those, because there aren't that many memory suppressor genes that have really been explored very deeply, even less is really known about, well, why is it that we should suppress these processes? And I think it's quite evident that from the data, like that it's not a one-off. Like it's not like we just have one or two memory suppressor genes. We have multiple, meaning that it is important to limit these processes. But exactly why is unknown yet.
Okay, we might have time for one more. Is there anybody else?
I'm not showing, but I'd like to ask a question. It's Brad.
Oh, Brad, go on. You're there.
Hi, and I enjoyed that. Very different from my area, but would like to follow up on what Ted was asking, I think, or maybe even comment now, because I thought you might go this way. I was wondering if there might be some advantage in suppressing memory if you're having to adapt to ever-changing environments. In other words, you don't want to pigeonhole yourself with a certain memory that would prevent you from adapting to a change in environment. And would there be, I would imagine you could probably find a way to test that. I don't know.
Yeah, that's definitely one of the disadvantages, I think, to having better learning and or memory. Is that, yeah, you do need your nervous system to be able to adapt to new stimuli and change, as you say, changing environments. And so, yeah, how these various memory suppressor genes, alter our ability to, forget these, prior associations that may not be true anymore, is also kind of an interesting area of study.
Okay.
All right, thanks everybody. Anna, I want to thank you. That was great. It was fantastic. You hit it just right. I have to say that I love it when somebody shows me a slide and says this was 5 years of my life.
Yeah.
Because we all have one of those, and sometimes it takes a little bit of imagination to get it all in one slide, and sometimes it's a testament to how hard you've had to work to reach some kind of an outcome. So I think that's really cool. I think we all have one of those somewhere, and yours is very nice. You will receive a much coveted Department of Biological Sciences water bottle eventually, probably in your mailbox.
Yay.
And with that, I want to thank you. Yeah, last point, thank you all for attending. Wonderful to see everybody. The only other thing is that there's a slight change in schedule, and next month's chair's lecture will be given by Rich Palmer. So it was going to be Thames, and Thames is going to be shuffled to January. We had to do a little bit of a schedule shuffle, but next month will be rich. So with that, I'd like to thank you all for being here, and thank you again, Anna. Great job, and everybody, take care.
Thanks, everybody.
Hope to see you all soon.
Thanks, Dave.
Yeah, that was good.
Yeah. All right.
Take care, man.
Take care.
Yep.